and find useful for their research. So there's a sign-in sheet going around. Please sign it if you haven't already. And today's speaker is Kraig Stevenson, and he's going to talk to us about best practices for quantifying allele-specific expression from RNA-seq data.
All right. Yeah, thank you, Marcy, for the kind introduction, as well as for inviting me to come speak. So my research, I'm a bioinformatics graduate student in Tricia Whitkop's laboratory over in the Department of Ecology and Evolutionary Biology. And so far, my research has largely focused on improving our estimation of both total and allele-specific gene expression from next-generation sequencing types such as RNA-seq, largely in the context of studying gene regulation and how this process has evolved differently both within and between species. Rather than focus on the biology of gene regulation today, I'm instead going to focus on what I consider to be some of the best practices for quantifying allele-specific expressions from RNA-seq data. So first of all, why study gene expression? Well, kind of at its core, this represents the biological process of converting an otherwise inert sequence of nucleotides into some functional gene product. And the gene product I'd like to highlight today is that of messenger RNA. This is a gene product that is transcribed and then could eventually be translated into a protein. So this kind of provides the most basic link from a genotype to a phenotype as a form of gene expression. And the way in which, so it's actually relatively easy to study individual genes' levels of expression, but a few years ago there was what I consider to be a revolutionary technology that was developed, allowing us to study the overall gene expression level of the expressed genome. And that technique was known as RNA-seq. And this technique proceeds usually in the following way. We have a particular sample of interest. This can be a model organism, a favorite model organism, could be any type of sample. mRNA is typically extracted from this sample. It's fractionated and converted into cDNA. We can't directly sequence RNA. We have to convert it to something that we can actually sequence. This is submitted to a sequencing machine, and at the end of this process, we're left with millions and millions of short strings of bases. And this is really where the bioinformatic challenge comes in. What's not so much anymore a bioinformatic challenge, but the main thing that we'd like to discover from these reads is their genomic origin. So one of the first things we might envision doing is aligning these short sequence reads to some sort of reference genome. And if we're lucky enough to have functional annotations for this reference genome, as you can see, there are different numbers of sequence reads aligning to these different functional annotations. And we can use the number of sequence reads aligning to these genes as a proxy for that gene's level of expression. And this sets up a very natural problem of, okay, once we've looked at gene expression in one sample, we might envision comparing this across different samples that differ by some sort of condition. They could differ by environment, really anything you could think of. But total gene expression isn't quite the end of the story. As we know, in diploid organisms, a gene's total level of expression is a result of contribution from both a maternal and a paternal allele. So in this case, I'm highlighting for the same example that I just drew out, a set of allele-specific reads aligned to this genome. So here, the challenge is differentiating our set of reads into each one of these alleles. So why might someone be interested in allele-specific expression? Well, one such example is we can use the output of allele-specific expression as a measure of the cis-regulatory output of a sample. So whether or not there could actually be base changes in cis-regulatory regions affecting a gene's level of expression. We can also use differences in allele-specific expression between samples that have identical parents that differ only by which parent contributes a particular allele. We can use differences in allele-specific expression between these samples to look for a phenomenon known as genomic imprinting. This is an epigenetic phenomenon whereby either the maternal or paternal allele is epigenetically silenced. So in this case, if we compare these two samples that again differ only by which parent contributed which allele, a blue or a red allele, we could see that in fact the paternal allele in this case is being silenced. So today I'd like to highlight for you how we can go from this type of framework, aligning allele-specific sequence reads to a genome, how we can do this accurately to quantify allelic abundance from this type of next-generation sequencing data. I'm also going to go through how to create appropriate genomes for mapping allele-specific sequence reads, and then ending with a test for significant differences in allele-specific expression for this type of data. So like the problem suggests, we really start by aligning our individual sequence reads to our reference genome. And when we align these reads to our single reference genome, what we can actually use are the base content of individual reads overlapping a particular position in our reference genome to look for some alternative allele. So in this case, for this particular position, I actually have two supporting reads overlapping this position that shows some alternative base, in this case T. In this framework, we must allow for mismatches to our reference genome to accommodate this alternative allele. And usually these type of alignment procedures are done enforcing unique alignment to our reference genome. No questions over there yet? Okay. In fact, when Degner and colleagues several years ago attempted this procedure, they found that when aligning sequence reads to a single reference genome, this in fact yielded relative measurements of allele-specific expression that were biased toward the reference allele. So let me lead you through this, one of the plots in their paper. So here they're showing for a panel of SNPs between two individuals, shown here in red and blue, the proportion of the reads matching the allele represented by the reference genome. In addition to a deviation from this expected distribution, shown here in the black dashed line, they saw an increase in variation of this distribution, as well as a slight shift in favor of calling a proportion of the reference allele greater than 0.5. So we really wanted to figure out why this was happening. When this work came out, it was a little bit unsatisfying that we didn't fully understand the reasons why this might be occurring. So in a simulation study, I actually simulated allele-specific sequence reads, and I used one of my favorite model organisms that we also use in my lab, Drosophila melanogaster. And the way in which I did this was I used the Drosophila melanogaster reference genome, and using homozygous genotypes from a different line of Drosophila melanogaster, from what's known as the Drosophila genetic reference panel, I used differences to this reference genome to define some alternative allele. So here I'm showing in blue the reference allele, and in red the alternative allele. And again, because we're interested in gene expression, I'm limiting my simulation to a list of annotated exons. And then I started by generating all possible allele-specific sequence reads from each of these regions. And I did this both in the forward and the reverse complement orientation to reflect the fact that RNA-seq is typically done in a non-strand specific manner. And you notice I've colored some reads in blue and some reads in red. These colors will appear throughout the talk. These represent reads that overlap a differentiating site. That is a site that we know can differentiate between both of these alleles. I've also colored some reads in black. These are reads that, while they don't contribute to allele-specific expression because they don't overlap one of these sites, the sequence is identical between these two portions, they contribute to a gene's total level of expression, but not allele-specific expression. And to really determine the genomic properties of what might be causing the aforementioned bias, I've simulated this data without any sequencing errors. And this also, what you might notice in this setup is that for each differentiating site, this sort of very naturally, we set up this expectation that the allelic abundance from our reference and alternative allele should in fact be equal. So the proportion of the reference allele that we should be able to recover is 50%. That'll become important in some of the later plots that I'll show you. So when we take this simulated data and attempt to align to our single reference genome, allowing a single mismatch, I found that there was around 50% of these differentiating sites that showed unequal measures of allele-specific expression. And that's what I'm showing here in this pie chart. If I instead plot the distribution of the proportion of the reference allele, as Degner and colleagues did, I see that, again, most of my differentiating sites that show unequal measures of allele-specific expression are in fact biased in favor of calling a higher proportion of the reference allele than expected. What we would expect is right here at 0.5.
So, Craig, let me just ask a clarifying question. In your simulated data, you know that it was 50-50.
Yes.
Okay, and so this is purely, it's not a result of the simulation, but of the aligning.
It's a result of the aligning. Yeah, and that's a further motivation for why we didn't incorporate errors into that simulation. Maureen?
So did you keep just one allele-specific difference in the region in your simulation, or are you using real genome data?
This is real genotype data. So in this, this is kind of a deployed example. Because here, this is a simple case. There's only one. Exactly.
But if you have like 4 in a read length, then maybe that's beyond your rate of acceptance of. Right. And that's kind of the punchline for what I'll show you after this, after allowing a single mismatch. So what Maureen said is very correct. There could in fact be individual differentiating sites that are closer to one another than the length of a sequence read. And I call these neighboring differentiating sites. So in this case where I've aligned, I don't know what that is. It's part of the font issue. That's good. When I align these sequence reads to a single reference genome allowing one mismatch, if there happens to be a read that overlaps 2 differentiating sites, this of course mismatches the reference genome by more than 1. And so all such reads that overlap more than one differentiating site would in fact be removed from analysis. They simply wouldn't be able to align to our single reference genome. So because of this, we hypothesized that increasing the number of mismatches would improve our estimates of allele-specific expression. And in fact, that was your question?
Sorry. Sorry, I didn't mean to cut you off. No, okay. So quick question, maybe I misunderstood this. So in the original analysis, you only allowed unique mappings, right? Because that's going to
still allowing unique
still
mappings
only allow
here.
unique mappings, but you're allowing unique mappings with a single mismatch, right?
Yes.
So how do you handle cases where you have maybe like gene families that are somewhat identical to each other that you might be mapping to another allele?
This is another issue that will come up a little bit later. So the issue Ryan's referring to is known as mappability, and I'll talk about how that can also have an effect or independent of mismatches. But yeah, you're right.
2 other questions. One, what are you aligning this with?
Bow tie.
Bow tie. The bow tie, not bow tie 2.
Bow tie 1.
Okay, and these are single end or paired end?
These are single end.
Okay.
Yeah, I've left off some kind of the
I'm just
experimental details.
There's actually some bias towards how, when you do paired end mapping, how it maps the paired end. It's not always as straightforward as you would think it would be.
Yeah, so these are all single end done with the original version of bow tie, which has a parameter that allows you to change the number of allowed mismatches.
Cool, thanks.
Okay, so as expected, when we increase the number of mismatches, we do see a broad improvement in our measures of allele-specific expression. So in the case where I've allowed up to 3 mismatches, I see roughly 10%, only 10% of the differentiating sites still showed some measure of unequal allele-specific expression. One thing we also hypothesized that as you increase, as these densities of differentiating sites increase, so might also the bias toward the reference genome. So the next step that we took was to stratify our differentiating sites based on these densities. And we wanted to observe how this actually affected our measurements of allele-specific expression. So I'll guide you through this plot really quickly because I'm going to show several of them. So on the y-axis here, I'm showing the proportion of the reference allele. And again, what we expect from the simulation is that this should be balanced at 0.5. We should be able to recover half of the reference allele, the other half being the alternative allele. And on the x-axis here, I've stratified the differentiating sites based on their density. So a density of 1 in this case indicates an isolated differentiating site, one that has no neighbors within the length of the sequence read. And if anyone's interested, the length of sequence read here was 36. What you immediately see is that as this density of differentiating sites increases, so does this bias toward calling a higher proportion of the reference allele than was simulated. And again, because we've also allowed additional mismatches, we want to look at how this also affected differentiating sites when we separate them into their relative densities. As you might expect, for higher densities, this also caused an increase in the bias toward preferentially calling the reference allele. One thing you'll notice, though, is that when allowing a single mismatch, 2 mismatches, and 3 mismatches, we have these relatively, what should appear as box plots actually appear as lines because the data are so densely populated around 0.5. These are cases where, for example, allowing 3 mismatches, we've allowed an appropriate number of mismatches to accommodate neighboring differentiating sites of up to 3, up to and including 3. And this also holds true for 2 and 1. So that explains why in these situations, the systematic bias toward either allele has been removed. So as an alternative strategy, we can use, if available, we can also use independent alignments of our sequence reads to both a maternal and a paternal genome. This is actually a strategy used by an allele-specific mapper known as allele-seq. And the way this works is we take each of our sequence reads and independently align them to each of the maternal and paternal genomes. Then ask for a sequence read, did it uniquely align to one or the other genome? So in this case, this read allowing no mismatches, because here there's really no need to allow mismatches to accommodate some reference allele. That's represented by the genome, the allele-specific genome itself. So here we would see that this read, in fact, aligns uniquely to the maternal genome, but because of this mismatch, would not align to the paternal genome. We use that as evidence that a read is coming from the maternal genome. And similarly, reads colored in black are those that align equally well to both genomes. And so, again, these contribute to a gene's total level of expression, but not allele-specific expression. And here I'm also enforcing unique alignment. So when we use this strategy, we see that regardless of the density of differentiating sites, the systematic bias toward preferentially calling, in this case, the maternal allele, arbitrarily chosen, has been removed. And the proportion of differentiating sites showing some level of unequal allele-specific expression has been drastically decreased. Okay, so now I actually want to draw your attention to the issue that Ryan was mentioning. So even in our best case scenario, aligning to a single reference genome, allowing 3 mismatches, so in these cases we've allowed an appropriate number of mismatches based on the density, this actually doesn't affect the strategy of aligning to both maternal and paternal genomes, but we still see differentiating sites that show inaccuracy in their recovered measures of allele-specific expression. So one thing that we hypothesized could explain this are differences in mappability, and that is our ability to uniquely map, because that's one of the criteria that we're enforcing, are differences in our ability to uniquely map these reads to a reference genome. So here I just want to give you a couple examples of how mappability could be affecting this. So in one case, I'm showing a gene paralog that's it's related to the example you were highlighting, right? So in our reference genome, we have this gene paralog that has identical sequence information, making it impossible to determine, using a unique alignment, the genomic origin of this read. However, in this alternative genome, because of a couple base changes here between these two paralogs, we can fully distinguish any reads overlapping these two base differences from this other copy elsewhere in the genome. Another issue that can affect genome mappability are regions of, that didn't line up properly, but that's okay, are regions of low sequence complexity. So in this case, I'm highlighting in the alternative genome a small microsatellite, a GA repeat region, making it also impossible to determine the exact origin of this particular sequence read. However, in the reference genome, this area of low sequence complexity happens to be broken up by some base changes, making it possible to determine the genomic origin of this particular read. So we then took our differentiating sites from our different methods and classified them as either showing perfect or imperfect mappability to our particular genome. So in our single reference genome case, where we're allowing a single mismatch, 4 differentiating sites labeled here in white that show perfect mappability, we were largely able to recapitulate the simulated level of 0.5 proportion of the reference allele. However, for differentiating sites showing imperfect mappability, here in gray, we saw a much more variable distribution in this proportion of the reference allele. This was also the case when aligning to a single reference genome, allowing 2 and 3 mismatches. Also, not entirely surprising, this explained almost all cases of unequal allele-specific expression in our strategy where we're aligning to both maternal and paternal genomes. So here again, I'm separating differentiating sites into those that show perfect mappability to both genomes, or those that show imperfect mappability to either genome. And as you might have guessed, those showing perfect mappability, again, we were able to recover almost 100% the simulated level of the proportion of the reference allele. However, for those showing any level of imperfect mappability, those again were much more variable. And what I'm not showing here, but was something we also looked at, the direction of this bias was directly related to which genome, either the maternal or the paternal genome, showed the level of imperfect mappability. If they were both, if both genomes showed imperfect mappability, then these measurements could appear anywhere. However, if only, say, the maternal reference genome showed imperfect mappability, we're more likely to see estimates of the proportion of the maternal allele in this area. We're preferentially calling the paternal allele, which showed perfect mappability to its genome. And one other question we wanted to answer was, after removing differentiating sites either based on their density of differentiating sites or based on any level of imperfect mappability, how many would we still be able to measure? And we also wanted to look at how many exons retained at least a single one of these differentiating sites. So in fact, in our, for lack of a better phrase, worst case scenario aligning to a single reference genome, allowing one mismatch, we were able to still use slightly less than 50% of the differentiating sites, but just over 80% of exons still had at least one differentiating site that we could measure allele-specific expression from. And of course, in our best case scenario, aligning to both reference genomes, almost all differentiating sites as well as exons were usable. And these small black bars indicate the additional contribution that imperfect mappability made to the proportion of either differentiating sites or exons we were able to measure. So this also highlights the fact that while mappability is an issue, it's actually a relatively small issue compared to that of the density of differentiating sites when aligning to a single reference gene.
So Craig, this again is with 36 base pair reads?
Yes.
Did you create something similar with 100 base pair? Because
not with 100, but I did.
With mappability, 90% of it would probably disappear.
Yeah, so mappability issues with 50 base reads do decrease, but are still present. As you also might expect, by increasing the read length, I'm also increasing the number of differentiating sites that could possibly fall within the read length. So that also increased the bias in the single reference genome case. I also looked at, so this was all done within Drosophila melanogaster. I also looked at a between species comparison to look at the effects of sequence divergence, and that also had a similar effect as increasing the read length. But those are results I'm not showing today.
So these are pretty different strains of melanogaster?
So the two species that I use, this. Are you, so the,
Well, some
like
stuff's
how
gone
polymorphic were they? Or?
Well, yeah, some stuff's gone through labs and it's like clonal almost.
These are pretty
Right.
close. These are pretty close, but not close enough to preclude being able to quantify allele-specific expression from. But close enough where for every genotype I had the corresponding base in the reference genome. No indels. Okay.
Right, for these areas of imperfect mappability, you said you've excluded them from this graphic here. You've excluded
sorry,
biases or not?
I've excluded them only up to this point, and these
Oh, I
black
see.
bars
Yeah.
represent, had I ignored mappability, this is how many I could have measured. So it's kind of a...
Is the imperfect mappability predictable? So could you create a mask for this, for the imperfect mappability, based on a set of rules or criteria?
Yeah, so I actually used a program called GEM to calculate genome mappability.
Okay.
And inputs to that program are the read length and the specified number of mismatches. The way it works, I'm pretty sure it simply generates all possible reads from the genome and attempts to align them back to that genome. And then for each site, I have a measure of how mappable a read stemming from that location of a specified read length,
Okay.
how mappable that read is.
Okay.
GEM is the name of that gem. Okay, so what I'd like you to take away from these results is that the density of differentiating sites increases the bias in measures of allele-specific expression when aligning to a single reference genome, and that the preferred method to reduce this bias, or in fact eliminate it, is to align to both maternal and paternal genomes, and that while imperfect mappability has a small effect, it's still a measurable effect when aligning to both maternal and paternal genomes. So now I'd like to tell you a little bit about how we can create appropriate genomes for this type of approach. So the way in which I've done this is we actually take a maternal and a paternal sample of genomic DNA and align them back to our reference genome. I realize what I just told you is almost in violation of what what I consider to be a best practice for quantifying allele-specific expression. Here, we're not nearly as concerned with recovering a particular allelic abundance as we are with just getting a good set of confident reads overlapping a particular site that shows some evidence of an alternative allele. So here, the relative allelic abundance doesn't matter nearly as much.
So is there an advantage to aligning to, when you say reference, do you mean the entire reference or a transcript reference? Is there any
Genomic DNA to the whole reference, or?
Oh, you know what? Sorry, I'm thinking RNA still.
Okay. Yes. Yeah, so this is basically to build a genome, but not in a de novo type of way. More like
Couldn't you do that from the RNA-seq data itself?
You could, but I would be afraid of differences. So there is an issue known as RNA editing that I think could give you a lot of false positives in your variant calling. So I would be a little bit more hesitant in directly using the RNA-seq reads to call variants. I think the superior approach would be genomic DNA samples. If
Sure, but sometimes you don't.
Sometimes they're not available, right? There was, no, never mind. That was a review paper, not one that I can talk about. What?
There is one approach for calling, at least one published approach for calling variants from RNA-seq data, but I don't know how widespread it's.
Okay.
I assume it takes the 8i type of editing.
Yeah, I imagine such an approach is the most successful when at least cross-referenced to a list of known variants that I have seen. But there you're still dependent on there being a set of known variants, which is also not always possible.
Do you do like an exon capture here? Because I would imagine that the number of reads you need to cover the Drosophila genome is pretty high.
I think with a single lane of genomic, a single lane of sequencing for genomic DNA, we can recover something in the area of 50x for the Drosophila genome. Yeah, yeah. So that's not an issue. But you're right. For other genomes, this might be prohibitive. So in reality, this data looks a lot more something like this. This represents a snapshot from the integrated genomics viewer showing individual read pileups of genomic DNA to this reference genome. So here I'm showing the maternal sample of genomic DNA as well as the paternal sample of genomic DNA. And in addition to these gray peaks, which simply represent for an individual site the number of overlapping reads, you'll see some peaks have been colored differently from this gray color. These represent elevated proportions of some possibly present alternative allele that this program is trying to call. Of course, we can't simply look at genome viewers to call our variants. We need a more robust approach that not only takes into account the proportion of reads representing an alternative allele, but also mapping properties of those reads overlapping this alternative allele. So one such approach that I've started using is known as GATK, the Genome Analysis Toolkit. And this is a highly customizable sort of workflow for calling variants from a set of sequencing data. So what I'd like to highlight with this workflow here is that level of customization, which can simultaneously be somewhat of a headache, trying to determine what are the best parameters for my particular sample. But in a lot of cases, if those are tuned well enough, you can get really, really good results from this variant caller. And in the end, you're either left with a sad face, that's a good set of variants, or sorry, a bad set of variants that you probably have to troubleshoot, or a smiley face set of variants that you might be able to use in your project. And this workflow also represents GATK's best practices for variant calling. That's where I stole my title from. So this, again, this degree of customization also highlights the sort of stepwise variant calling that GATK goes through. In particular, these individual modules that are calling variants or kind of making changes to the data are known as walkers. And they proceed through the data to perform various steps, a couple of which I'd like to highlight for you today. In particular, how the initial set of variants are determined. The real novelty of GATK is its implementation of a local indel insertion or deletion realignment, a base quality score recalibration, but this I actually won't go into much more detail on, as it requires a set of known variants, which is not always possible in every sample. That's maybe one of the drawbacks of GATK, although they do provide recommendations for working around this issue, as well as how our final set of variants are eventually filtered to get the highest quality set. So, sorry, that shows up a little bit grainy. So the first step is to identify our set of candidate variants from this type of alignment back to our reference genome. So in the case of a single nucleotide variant, the way that GATK works is actually a Bayesian formulation for individual genotypes. That is, the probability of a particular genotype given this alignment data is proportional to the prior probability of that genotype, this alternative genotype, and the likelihood of this read data given that particular genotype. We can talk afterward about the more specifics of that formulation for whoever's interested. As well as candidate insertions and deletions, I mentioned this is one of the novelties I consider with GATK, is its implementation of the Dindel algorithm, which actually does a local realignment of reads surrounding putative insertions and deletions to improve those calls. Because, as you might imagine, reads that are close to indels can have a higher level of mismatching bases to the reference genome. So this is an attempt to correct those mismatches and not have a spurious set of false positive variant calls. So once we have a set of variants, GATK also provides a set of recommended filters both for SNPs and indels. And rather than talk about the individual parameters, I'll just tell you a little bit about what these parameters hope to achieve. The first of which is a filter for the average read quality of reads showing evidence of the alternative allele. The second is actually using a Fisher's exact test for the detection of strand bias. And this is an attempt to reduce false positives. There's also a filter determining the consistency of a particular variant site, with there being 2 and only 2 haplotypes. Also, something taken into account is the average mapping quality of reads overlapping a particular variant. And finally, there's a small penalty for our alternative alleles that occur on the ends of reads. So for many sequencing technologies, the ends of reads are slightly more error-prone than the beginnings of reads. So variants that are supported by an unusually high proportion of such end reads, for lack of a better term, there is a small penalty associated with that. And finally, once we have our set of variants, we need a tool to incorporate them back into our reference genome. So if we consider this was our original reference genome, we input a set of high-quality variants. This is a file format known as VCF, variant call format. At the end of, or after inputting this information, we'd like to be left with a reference genome that has incorporated both single nucleotide variants, insertions, and deletions. This last case I'm highlighting actually in the metadata shows that this happens to be a heterozygous variant. So this is something in many of our analyses we would actually choose to mask. So instead of incorporating one of these variants randomly, we instead incorporate an N. as this can have drastic effects when using these genomes to quantify allele-specific expression. So the takeaway here is that we can align individual parental genomic DNA to our reference genome to call variants. We can incorporate those variants into the original reference genome to create maternal and paternal genomes. And these are genomes that we can use in kind of the aforementioned strategy to accurately quantify allele-specific expression. So finally, once we have a set of accurate allele-specific expression calls, how do we test for significant differences in allele-specific expression? Well, one of the first things that we actually need to do is normalize our read counts. And one of the more popular strategies for this normalization is known as the reads per kilobase per million mapped reads transformation. One problem with this technique, though, is that for a small fraction of genes that are either extremely large or very highly expressed, small changes in those genes can have a very drastic effect on more lowly expressed genes. So comparing RPKM transformed count data between samples can sometimes be problematic. And most statistical frameworks are tailored for looking at differences in total expression, not necessarily allele-specific expression. And I've given a subset of such programs: DE-seq, EdgeR, Poisson-seq, Bayes-seq, and CuffDiff. These also, a lot of these programs also are dependent on on the sample being replicated, which also isn't always a possibility. So in the absence of a sample being replicated, how can we compare these counts between samples? We actually need to use count statistics. Because we're using count statistics, these are very much dependent on the total sample sizes. And because of that, we need to actually perform what I'm calling downsampling. We need to actually take our set of allele-specific reads with more counts and actually reduce them to a smaller sample in order to make them comparable and to equalize the power for calling allele-specific expression between these samples. So in this case, if we imagine for a particular gene between two samples, we actually have evidence of twice the number of allele-specific reads in one sample compared to another. We can, it turns out we can use the hypergeometric distribution to draw n of these reads, so n representing the smaller, the number of allele-specific reads in the smaller sample. We can draw n reads from this sample without replacement and have a much more comparable set of allele-specific counts comparing these two samples. So is that kind of clear how this is working? If we had more than two samples, we would simply take the smallest sample and downsample all other samples to that size. And the reason the hypergeometric distribution works well for this is because this is the description of the hypergeometric distribution. We have a large set of objects, some of which are counted as successes, and we're actually drawing a subset of this larger sample size. And the number of successes in that smaller sample represents or follows a hypergeometric distribution.
So you're saying that you're throwing away data to increase some statistical power. That doesn't seem intuitive
to equalize the power between these two samples. Because otherwise, so estimates of allele-specific expression in this sample are more accurate than the estimates in this sample, just because there are more supporting reads. So it makes a direct comparison between these samples a little bit tricky because the power for detecting
that the sample size should be taken into account, like the read count here being the sample size in the statistical test. to measure differential allele-specific expression rather than throwing away the better sample and just dividing into the lab.
But throwing away a sample would sort of preclude and
Not throwing away the sample, throwing away reads.
throwing away some of this data.
So, Craig, I see what you're doing. I think it would help if maybe if you explain a little bit more your motivation, your goals. I'm not sure why are you comparing two samples? I thought the goal was to get to figure out which genes have an allelic imbalance for a single sample.
Right. So if you're, that's a good point. If you're just looking for allele-specific expression differences within one sample, you actually don't need to use this type of strategy. But sort of, if you wanted to compare for a single gene allele-specific expression differences between samples, these are not as directly comparable
Could
in
you do a
terms of
test
how.
that takes into account the difference in variation between the samples? You couldn't use DCMB or HR in that
Right.
case. But
And so the particular test we're using here is the binomial exact test. And I think because we're limited to using this test for this type of count data, I don't think we can take the variation
why are
into
you
account.
limited to that test?
I can't think of another test for
Okay, so for example,
comparing two alleles. I think this is kind of the gold standard for allele-specific expression of count data, where
I
you
would
don't
say
have
replicates.
replicates.
Someone could come up with a test that takes into account the difference in variability.
If
Allows
you have
to use
replic-
the data.
If you have replicates, none of this is going to But say you wanted to compare back
I
to,
haven't thought about it much, but.
say you wanted to compare back to some sample that was taken a long time ago that also doesn't have replicates, I think you'd still have to use these count-based statistics.
Well, it would still be a count-based test, but it could be one that takes into account heterogeneity and variance. I'll think about it.
Okay, yeah. It's not an easy problem. When you do this, it becomes an easy problem, an easier problem.
Right, yeah. I see.
But without doing this type of downsampling, I think it's really hard to compare between samples. And so the test we're using is the binomial exact test. We use this for each gene. And here we assume that our null hypothesis is that the maternal allelic count follows a binomial distribution of the total allelic count with a success probability of drawing a read from that maternal allele of 0.5. And the choice of this being the maternal allele is also arbitrary. This could have just as easily been the paternal allele.
So quick question, actually, just to, not to harp on this point, but if you have lots of samples, say 100 samples, right? And you have 99 of them that have a large amount of allele-specific data, right? And you have one of them that's got a smaller amount of data. Are you downsampling all of that to that small amount of data?
We've never encountered such an extreme situation like that. In that case,
Because usually there's always one or two samples that are.
in that case, it might, I hate to say this, but maybe you'd consider getting rid of that sample. That sounds bad. I know you don't want to throw away data. No one wants to throw away data,
Why
but
don't you upsample?
that,
That's cheating.
that's cheating.
It's easier to throw away data.
So throwing away data is not cheating.
But you can throw away data, you
No.
just can't grab data out of nowhere.
Is
So I was just
is there a
curious if
statistical
you
test for
handled
cheating?
that or if you ever came
Yeah,
across that.
we haven't encountered anything that's quite that extreme. So we've never had to say, oh, maybe we should actually throw this sample out.
And off of that point, because I have to ask, 'cause I'm a scientist, do you take into account, you're working with flies, so maybe this isn't as big a deal, but large enough population.
I've tried to make this independent of the fly work. I
Yeah,
hope
but
that's come
so
across.
You're going to have places where there may be gene duplications, right?
Yeah.
That are intrinsic to some samples that are others.
Mhm.
And so you're going to have more expression there that you're now downsampling further, right? So is that going to bias your results any since you're
So based on
downsampling a real effect?
based on how the allele-specific mapping is done, because we're also enforcing unique alignments, it's going to be harder to measure expression in those types of genes to begin with. Only because
But the unique
if-
alignment,
if those genes are so similar that you can't tell the difference between reads coming from this particular gene and its paralog, is that kind of what you're talking about?
Well, so
paralog,
I'm
or-
talking about polymorphic duplications that maybe aren't in the reference, right? So I have a duplication that you don't have that's not in the reference. So maybe it has a SNP or something in it, probably maybe not, depending on how far back in the lineage it went. But you might pick up that difference there, but you're going to see a huge allele imbalance in addition to an allele-specific expression imbalance. And so I guess I'm just wondering.
So that would that not be captured though in the construction of your maternal and paternal genomes and my maternal and paternal genomes? Those differences?
Well, if you
Handle that
if
too.
when you map the genomic DNA, you note it at that point,
Yeah.
yes. But you're just looking at SNPs and indels, not so much CMB. So I'm just wondering if you thought about maybe incorporating that kind of information along with the SNPs and indels, that hey, we have a duplication in some of these paternal at these locations, and so we expect an allele imbalance, and therefore some sort of an allele-specific expression imbalance.
Yeah. So far, I would say I haven't taken enough care about those types of structural variants.
I had to ask.
Okay.
It's going to change your allele frequency too.
Okay, so just to wrap things up, I hope I've convinced you that aligning sequence reads to both maternal and paternal genomes can give us the most accurate measures of allele-specific expression, and we can construct these genomes by calling variants from separately aligned parental genomic DNA to a reference genome, and that using downsampling and exact tests for count data are ways we can determine significant differential allele-specific expression in the absence of a replicated sample. So with that, I'd like to thank my advisor, Tricia Whitkop, as well as a postdoctoral researcher, now turned research scientist in the lab, Joe Kulan, as well as the rest of the Whitkop lab. And thank you all for your attention.
I think that you should look up into something which is called like tests of difference of proportion, which I think is exactly what you're testing. Maybe I'm wrong, but there are 9 of them all implemented in R, so you can use them like straight away.
So, difference in, like if you're comparing two samples?
No, the proportion of 2 things, like it's 30% in one sample,
Yeah.
30% in the other sample. Is there a difference? Well, that depends on how accurately the 30% and the 70% was calculated. And then that depends on the number of reads supporting either allele. So then the null hypothesis is that there is no difference.
Mm-hmm.
So the difference is 0. And then there are tests which try to
What if you had more than 2 samples?
I don't know.
We'll also point out that building separate maternal and paternal genomes works fine with inbred model organisms.
How do you know I like
Will
inbred
not
model
work
organisms?
in an outbred species.
Unless you have
Because-
each parent sequence individually.
That would work fine in my family.
The whole family.
You need haploids
Yeah.
from each parent,
Mhm.
because you don't have phase within each
You're right.
parent.
You're right.
Well,
So get the parents to donate
Well,
sperm and egg,
well,
and then I can
right.
do this test.
Right.
Okay.
You already mask any sites that are heterozygous. So you don't care. You only look for homozygous differences.
Explain.
So
But for those homozygous differences, like say you just had, so like what you're talking about is maybe a hybrid from the wild whose genomic DNA you of course can sample, right? Aligning that to a reference genome, how do we then call our individual
No,
haplotype specific?
if you've collected 2 hybrids from the wild and mated them, there's still a question of phase within each of the parents. Because each of the parents is out
Guess
there.
why would you? Or why would you make?
I'm trying to make the analogy to RNA sequencing of humans,
Okay.
where each of the parents is outbred.
I think you just use the homozygous differences between them. The difference is that in the
But wouldn't
outbred
phase
parents,
still matter then? So you can still determine what are the homozygous variants, but which homozygous variants go
No, but there will be
together to
very
construct the habitat.
few
So in those instances,
variants homozygous
Yeah, then-
within either parent.
So then you're trying to get allele-specific expression from a situation where you don't know any allele-specific information. Like, it's not gonna work. That's.
You're being too perfect.
Also, there are
He has that
14,000
fault.
at least pseudogenes in the human body, which adds to this problem.
One more question out there. One more for, I mean, it's not really a question. So you focused on the technical aspects of this, which is tools and tech. That's what this is for. It's really cool. Just a quick question about application. So are you guys using allele-specific expression as maybe a proxy for isoform expression, where you have alleles in particular splicing structures, and you can show differential splicing with that?
This comes up almost every time I talk.
That would be really
We
cool.
actually aren't, but our collaborators use, we're using the same set of data. Our collaborators, Brenton Gravely, who is part of the Modern Code project for Drosophila, is actually using this data to look at differences in regulation between splice isoforms.
This seems like it's
Yeah.
really cool.
It's sort of not my lab's area of expertise, but someone's using this data exactly for that.
Also, one idea that might be crazy, I didn't really think it through, but you're solving the problem of allele-specific expression by first aligning to the genome. Well, maybe you don't need to align to the genome. Maybe you could like make allele-specific differential expression without alignment at all. Have you considered something like that?
So sort of counting
Counting
Kamers.
maybe, or other hashes of the possible.
So yeah, Kamer counting could maybe work for that. Of course, assuming you can map those Kamers back to their location in the genome.
But then the Kamers would be like masked on the position where the alleles differ.
Yeah, I'm trying to think how that would
Or locals.
work. You might be able to do something like that, independent of reference genome alignment, maybe. I'd have to think about that a lot more. Yeah, that's an interesting thought, though. All right. Thanks again, everyone.